Here's a short video about the discovery of vaccines.
Ten thousand years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected, but it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairymaids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family. But when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the Buddhist nun, dairymaid Sarah Nelms, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
So there's some pretty amazing things in that story. One of them is that this occurred around 1796. This was before people knew that microbes caused infections, so they didn't even know why this worked. They just knew it did. The other one was Jenner's doing all this work with smallpox and he didn't get smallpox, but apparently he was variolated as a child, so that probably protected him. So if you look at your um, learn your lecture study guide, there's the word vaccine. But first, let's do the word variolation. Remember, the word variolation was the technique they used to put the ground up smallpox scabs into somebody's, they blew it in their nostrils. And they called it variolation, but you had the risk of getting smallpox by that. So Jenner came up with using cowpox and did a technique called that we eventually called vaccination. It turns out vacca is Latin for cow. So even in the name of vaccine, the history is built into that word. So hopefully you got the connection between smallpox, cowpox, and Edward Jenner. But remember that this was a huge deal because smallpox is a horrible disease, and like I said, people didn't even know why it worked. They just knew it worked. Here's an amazing picture that I came across. 1901 photograph of an unvaccinated boy with next to a boy that's been vaccinated for smallpox. So pretty amazing. Apparently, when you get smallpox, I think you have like a 30% chance of dying, and this appearance that you have on the outside, you also have that kind of disfigurement on the inside. So it is painful. It's disruptive to all your body systems. It's just a terrible, terrible disease. It is the one disease that one human disease that we've been able to eradicate. So, people like me that are older still have a scar. I still have a scar on my arm for a smallpox vaccine, but probably most of you guys don't because you're much younger and um, we stopped giving vaccines at that point. So, Edward Jenner, 1796. Okay. So here we are. Name the 4 most common types of vaccines, what they are made from, and some of the pros and cons of each. So again, this is a table in your book. Let me see. It's table 1311. So what I did is I just looked at 2 different parts, put the 2 different parts on 2 different slides. So if we look at the 4 types of vaccines, they're going to be the killed vaccine, or you might hear this called inactivated. I says viruses are termed inactivated instead of killed. The live attenuated vaccine, and then there's subunit vaccines, parts of organism, and a toxoid vaccine. So let's talk about each one of these. So the first one, let's go to the killed vaccines. So what was done with this is a pathogen is taken, whether it's a bacterium or a virus, and it's killed. You can use chemicals. Here it says by treating them with chemicals, radiation, heat, or some other agent. But it doesn't destroy its antigenicity. Remember, the antigens are proteins on the surface of the cells. So that there's that cell. It's the active pathogen. It's heated or treated with chemicals. There's lots of science that goes into trying to figure out what to do. And now it's dead, so it can't cause disease, but it is, and it's given to as the vaccine. So this is the whole microbe, but it's killed. And there are some advantages. That you have, all the antigens associated with that pathogen. So there, lots of antigens to stimulate the host and to stimulate those B cells and antibodies to be made. Here it says, let's see, it sometimes can give long-lasting protection. It might require fewer doses than other types of vaccines. But the thing to be aware of is this just doesn't work for all pathogens. So this is one of the techniques that you use killed vaccines. The other one is using the whole bacterium or the whole virus, but you don't kill it, you just weaken it. And that's called live attenuated. Attenuated means weakened. Contains live microbes whose virulence has been attenuated, which is weakened or eliminated. And it says it can be achieved by modifying the growth conditions or manipulating microbial genes in a way to make it less likely to cause disease. So things like the MMR and polio vaccines are live attenuated. These actually are some of our best vaccines against viruses, because what it can do is it can cause you have a weakened virus, so it can't make you sick, but it can multiply inside of your cells. So live attenuated viruses can cause a very long-term and a very powerful immunity. You're going to have this virus in your body for an extended period of time, and so that can continue to stimulate your immune system. It might even give you lifelong immunity. So some of our most effective vaccines have been live attenuated. The disadvantage is, here it says they require special storage facilities, but also if you have a live virus, it could potentially mutate and become virulent again. Now, this doesn't happen very often, but there are some notable examples when it has. So it's not likely to happen, but it could happen. The other two vaccines, the subunit vaccines, so these are just parts of an organism. So here's a virus, and it's just looking at the spikes on the virus, or which are acting as antigens, or whatever part, the toxin or the proteins on the outside of the bacterial cell. Here they have show some proteins that are in the flagella of the bacterium. And so for some pathogens, just the pieces of the virus or the bacteria can actually cause the immune response. And again, it doesn't work for all vaccines. And in fact, these generally don't give as strong of an immune response as the whole bacterium or the whole virus gives. But these work for some diseases. So these would be subunit vaccines, just the antigens from the surface. And then the last of the four major types is a toxoid vaccine. And the toxoid vaccine, some bacteria, actually the bacterium itself isn't what makes us sick. It's a toxin that's produced by the bacteria. So the classic one I can think of is tetanus. And so when you get tetanus, tetanus is you get a cut and you get tetanus bacteria in there. The bacteria produce tetanus toxin, and that toxin is a neurotoxin, and it causes your muscles to contract and stay that way. So the tetanus vaccine uses the toxin and not the bacterial cell at all. And it weakens the toxin, so it's an attenuated toxin or it's a weakened toxin, and you make antibodies against that. And so it's a special type of vaccine. A toxoid vaccine is a weakened toxin. So we take a weakened tetanus toxin, put that in our bodies, and we make antibodies against it. So those are the four major vaccines. Toxoid vaccines work pretty well. We know we have to get booster shots of those once in a while, but they work pretty well. But not all bacteria make toxins or viruses make toxins. So you can't make these for all pathogens. This is not in your book. In fact, your book mentions DNA vaccines, but it doesn't mention RNA vaccines. And it turned out that mRNA vaccines, messenger RNA vaccines, is a new kind of vaccine and became very, very important to fighting the COVID-19 pandemic. And so I wanted to add a fifth type of vaccine to your major types of vaccines, explain how mRNA vaccines work and the advantage of this type of vaccine. Well, during the pandemic, one of the big things that allowed us to, that pushed companies into trying to make mRNA vaccines is because you can make a lot of it fast. These other vaccines, there's different things like growing up virus when you're trying to make a whole vaccine, or just to make the subunits. Subunit vaccines are just generally hard to make. You just have to get lucky for them to work. The the killed vaccine or the live attenuated vaccines, it takes a long time just to grow enough virus to make the vaccine. But they hadn't even figured out that the vaccines work yet, and they still hadn't even started to have time to make virus, to grow virus up. So the mRNA vaccines can be produced a lot faster. You can go from no vaccine to a vaccine in hand a lot faster. And that was really critical during the COVID-19 pandemic, because people were dying. And we wanted to be able to get vaccines into people to stop this pandemic. So the mRNA vaccines turned out to work really well. Most of us got a few of them. And so here it is. They built this little kind of lipid bubble, and they put some RNA in there. And the RNA, remember from your genetics section, RNA is the instructions on how to make a protein, right? DNA to RNA to protein. So they put this RNA in here, and the RNA is the instructions on how to make the protein. This lipid bubble fuses with the membrane of your host cell. This is your own cell, the host cell. And the mRNA goes in there. The mRNA has the directions on how to make a spike protein on the virus. And so now we have translation, and you make a protein that looks like a spike protein. So with the mRNA vaccines, you're actually having, you're making virus spike proteins in your own cells. And what your cells do is they put these spike proteins on the surface, and you make antibodies against them. And so you can mount an immune response to spike proteins. So when an actual virus enters your body, these antibodies will recognize it. There was a lot of discussion about will these mRNA vaccines incorporate into into our chromosomes? And this is the reason why they used RNA instead of DNA, because remember, our chromosomes are made of DNA, and the RNA won't go in the nucleus even. So that's not a problem. That's not a concern to have. But this is the mechanism for the Moderna and the Pfizer vaccines that a lot of people got, the mRNA vaccines. I think I'm going to stop right there and then do the last part on another video.